Hi everyone and welcome back to Biostat Squid. I'm really happy to be back and bring you this tutorial. So in this tutorial we will go over the basic steps of preprocessing for single-cell RNA-seq data in R using the Seurat package. And we will go step by step through the standard preprocessing workflow, including basic quality control measures, normalization and scaling, and clustering steps. And we will do this using the Seurat package, which is very common in transcriptomic analysis. So let's dive in. Okay, so before we start, I'm going to show you where to access the dataset I'll be using for this tutorial, so you can download it and use it as well. But of course, you can always use your own single-cell RNA-seq dataset. So what you need to do is access 10x Genomics and go to a part called Datasets. And you will see you have many different types of datasets available, obtained using different sequencing methods, and we're particularly interested in single-cell gene expression data. And we're going to be using the non-small cell lung cancer dataset from 7 donors. So you can click on it and you will see you have different files you can download with different kinds of information. You can also read about the experiment and the sequencing method itself. And the file we're interested in is this HDF file with the raw gene expression data. So we can download it by just clicking on it and you have your data just there. Great, so now that we have the dataset, we can actually start coding and start our analysis. So just so you know, you can also download the data from biostatsquid.com. And of course, you will be able to find all the links and the code in the description down below. Right, so let's start. Okay, so before we start, it's always good practice to clean up our environment in case we have hidden objects in the background, and also to free up memory and to set our working directory. So we're going to do just that. So first we will clear all objects, including hidden objects, free up memory, and set up basic OR options. Now we have to load the relevant libraries. In this case, I'm going to load the tidyverse library, which I think we don't really need particularly in the script, but I always find it useful to have it from the start. And of course, we're going to be using the Seurat package. You might need to install it if you haven't done that already. Right, so now we set our input path and we set our working directory to our path. So the path is basically the location of your file. It's where R is going to look for the data. And it's also important to know that every time you save something from R, it will also save it to this working directory unless you specify otherwise. And we can check the files we have in that path. And as you can see, we have our data, the data we downloaded from the 10x Genomics website, and the script. Right. Now, I also like to set seed. Again, I don't think we really need it, particularly in this script, but it's always nice to have just in case you forget. And it just ensures that you get the same results when you use a randomization step. So for example, if we were to select random samples from our dataset, this ensures that every time you run the script, you will select the same random samples. So others or you in the future can reproduce what you did and your results. So again, in particular, in the script, it doesn't really matter, but I'll just leave it there. So now that we're good to go, we can start by loading our data. So I downloaded our data set as a .h5 file. You can see it here, and we can read it in using the read 10x h5 function. I will store it as non-small cell lung cancer sparse matrix data. And we will go into sparse matrices in just a minute. Perfect. And it has loaded now, and as you can see, it says genome matrix has multiple modalities, returning a list of matrices for this genome. So what are these multiple modalities? So we can check the structure of this object, and as you can see, we have a list of three gene expression or matrices. One of them has the actual data from gene expression, which is the one we're interested in. There's also data from antibody capture and multiplexing capture. So again, in this for this tutorial, we will just be using gene expression data. So that's the data we're interested in, and we will then assign it to an object called counts, CTS for short. And here we have our count matrix with the counts from the single cell RNA-seq sequencing. And as you can see, it's this matrix is of type DGC, and it's a sparse matrix. What does a sparse matrix mean? So basically, a sparse matrix represents zeros, so when no molecules are detected, as dots. So I can just quickly show you how that looks like. So I'll just randomly subset my object. Let's choose, for example, rows 1380 to 1400. Of course, this is all random. Let's see if this works. Let's say 380. So now we're just subsetting our counts object, and as you can see, we have the genes. So each row is a gene, and each column is a cell, a sample. So as you can see, most values correspond to zeros, and instead of zeros we have dots, and this makes the object much smaller. So it's everything is much faster, and it takes up less memory. And as you can see, values that are so non-zero values actually have the value itself. You can, by the way, also access particular genes. So say, for example, if we wanted genes, I don't know, maybe the ones we have over there, and let's say I'm trying to find a nice one, this one here, it will also print us out these values. Of course, only 30 samples. Anyways, let's continue. So this is our gene expression data, our counts data, and now we're going to convert it into a Seurat object, which is the object we're going to be working with in this tutorial. So we're going to create a Seurat object with this raw non-normalized data. And we just have to specify where the counts matrix is, so that's our CTS object. We can give the project a name, and then we can also select minimum number of cells, a minimum number of features. And in this case, we will only include cells with at least 3 features, so cells that have at least non-zero values for 3 genes, and features or genes that are expressed in at least 200 cells. Right, so we have our Seurat object here already. We can click on it to have a look. This is the basic structure, and I will not really go into details of the structure of a Seurat object, but if you'd like me to create another video explaining the structure and how to move around or edit a Seurat object, I'll be happy to. Just leave me a comment in the comment section down below. Squidtastic. So now we can start our pre-processing steps, and these steps might seem a bit boring, but they're absolutely essential if we then want to draw proper conclusions from our data. So the first step is quality control, or QC. What does this mean? So essentially we want to remove cells with a very low number of unique genes detected, as they probably are low quality droplets or empty cells. But also cells with a very high gene count, as it probably means they are doublets. So sometimes cells stick together, and then you're getting gene expression data not from one cell but from two. And another good quality control measure is the percentage of mitochondrial genes. Why? Well, as you probably know, mitochondrial genes are transcribed in the mitochondria, not the cytoplasm. So if we detect a high number of mitochondrial genes, it means it's a low quality cell or a dying cell. Perhaps the mitochondrial membrane broke, or there is some kind of apoptotic process going on, and the mitochondrial transcripts are leaking from the mitochondria. So in short, we don't like cells with a high percentage of mitochondrial genes. And we can start by calculating this percentage for each cell. And we can do it really easy with this function, percentage feature set, which is a function from the Seurat object, which allows us to calculate the percentage of features with a certain condition. In this case, we want to count mitochondrial genes, so genes starting with MT. So here we have some examples of gene names, and for human genes, mitochondrial human genes start with MT. Capital MT, by the way. If you're working with a different species, so for example mouse, mus musculus, I think it's lowercase letters, so you have to be careful with that, as it can change a bit. Anyway, we will just calculate our percentage of mitochondrial genes, which is now stored in the metadata of the Seurat object. And we can access it by using this at metadata. So we can just access the view, and as you can see, we get this data frame with where all the metadata of your Seurat object is stored. And you see there are already some columns we didn't create. By creating the Seurat object, it automatically will add n-count, which is the total number of molecules detected within a cell, and n-feature, which is the number of unique genes detected in each cell. So basically a low n-feature, so low number of unique genes detected, will indicate that that cell might be dead or dying, or it's an empty droplet. You're not sequencing enough genes, basically. A high number of counts, so high number of molecules detected or high number of unique genes detected, might actually indicate that this cell is in fact a doublet or multiplet. So it's for quality control, we might want to visualize all these metrics, and we can use this function, which can plot all the metadata columns we want. And all these metrics, it's important to visualize them because we can see how good or bad our data is, and use this to decide which thresholds to use to filter the data. So for example, here we see that there are quite a few cells with a high mitochondrial contamination, and we will need to filter these cells out. Another nice way of visualizing this quality control metrics is by drawing a regression line. So basically by plotting one against the other. And with this function, feature scatter, I will represent the number of genes against the number of molecules, and also I add this geom smooth to get this diagonal line. So let us just zoom this up a bit, and we can see that each dot is a cell, and we're representing the number of molecules detected in each cell against the number of unique genes detected in that cell. So most cells should follow kind of a linear pattern. We see that it plateaus a bit towards the end. And what we do not want to see is cells in this lower right corner, because this would mean that they have a high molecule count but low number of unique features. So we're basically capturing a very low number of genes and just sequencing those genes over and over again, giving a high number of transcript counts. We should not see cells in the top left corner of the plot either, since that would mean that we're capturing a high number of genes, but they are not deeply sequenced enough. So you should always check for this and make sure that this is due to low quality cells that you can filter out, and they're not due to a sequencing error or some kind of artifact. And if they are there, you need to remove them, as they will just mess up your data in downstream analysis. So how do we filter out all these kind of cells? So we can do this by using the subset function. And for this standard pipeline, I will just filter cells that have unique feature counts over 2500 or below 200. So they will be filtered out, and cells that have a higher mitochondrial percentage than 5% will also be filtered out. Of course, feel free to adapt your thresholds to your project. That's why it's always nice to visualize the different quality control metrics to see what makes sense in your dataset. Nice. So after removing unwanted cells from the dataset, the next step is to normalize the data. And by default, we will employ this global scaling normalization method, so log normalize, that normalize the feature expression measurements for each cell by the total expression. And it multiplies this by a scale factor. The default is 10,000, and it then log transforms the result. So these are the default parameters. So even if you just run normalized data, this Seurat function, it will use this normalization method. And we can see the normalized values, which will be stored in the at data. Where is it? Here you go. They will be stored here. Nice. So we can next calculate a subset of features, so a subset of genes that have a high cell-to-cell variation in the data set. So genes that are highly expressed in some cells and lowly expressed in others. So they can be used to distinguish a certain subset of cells from the others. And it has been found that focusing on these highly variable genes in downstream analysis actually helps to highlight the biological signal in single cell datasets. So we actually focus on what the actual biological variation, the actual important data that allows you to distinguish between a subset of cells. And by default, this method will return 2000 features per dataset, but of course you can always change this. And we can actually have a look at the top 10 highly variable genes by using the head function. We can also kind of plot them with this variable feature plot and label the points. So here we have, and these are in red, we can see our 2000 highly variable genes, in black the less variable ones. As you can see, we're representing standard variance here, and we can see the labels for the top 10 genes. You can also change this. So once we found the variable features in our dataset, the next step is to scale the data. And this step is used to remove unwanted sources of variation. For example, technical variation introduced during the experiment, such as batch effects, or biological variation, such as variation due to the cell cycle stage or mitochondrial contamination. And we can correct these by scaling the data. So then when we cluster the cells later on by similarity of gene expression, we can be sure that they are clustering due to the actual differences in gene expression and not these other unwanted sources of variation. And if you want something a bit more complex, you can also check Seurat's new normalization workflow with the SC transform. But for the sake of this demonstration, we will stick with the standard parameters and just use the scale data function. Nice, you can also view the scaled data here if you access it through assays. Nice, so we finished all the normalization and scaling steps. Great, so now we can move on to dimensionality reduction, which you might find a bit more exciting. And well, dimensionality reduction methods allow us to visualize our multidimensional data set. And one of the most common methods is PCA, which we can compute using the run PCA function. And if you're not very familiar with PCA, you can check my other video, but we're basically summarizing all the gene expression data across many genes into principal components. So each cell can now be defined in a way by these principal components. So the function might take a while to compute depending on how big your data set is, but eventually you will get your results. So basically for each PC you will obtain loadings for each gene, which tells you how much does that gene contribute or explain the variation in the data set. And remember that principal component 1 explains more variation in the data set than principal component 2, then principal component 3, and so on. So the function will print out the top genes that contribute positively and negatively to each PC. If, for example, you want to print out the 5 top contributing genes to each PC, you can do it using this print function and set the number of features to 5 in this case. And well, another way of visualizing the PCA loadings is with a heat map, which looks something like this. And in this case, we're seeing the loadings or the scores for principal component 1, and we're just looking at 500 cells. And we see some of the genes, some of the top genes contributing to that principal component. And as you can see, this is a nice way of, let's say, grouping cells according to this principal component, which takes into account gene expression data across multiple genes. So now we're not really looking into particular genes, but each principal component is actually taking into account the information or the gene expression data of multiple genes. So perhaps the best way is to visualize the first few principal components. Let's do this with this function, dim plot. And this is an example of a PCA plot. In this case, we're representing PC1 against PC2. And each of the dots is one of our cells. And they are placed according to the PC1 and PC2 loadings, which again take into account the gene expression data across all the genes in the data set. And we can see the cells group in certain clusters according to these PCs. So as an example, this group over here might have a high expression of a certain subset of genes A and low expression of a certain subset of genes B, whereas this other group here will have a low expression of that subset A and a high expression of that subset B. So as you can see, principal component analysis is a great way of visualizing the whole data set, so all your cells, taking into account the gene expression data across all the genes in your data set. Nice. So as you know, the run PCA function returns the loadings for many different principal components. Now, how many principal components do we need to explain the variation in our dataset? Are 2 components enough, or do we need more? Now, there are many ways of checking for this, of deciding how many principal components explain enough variation in our data set. But one of the most straightforward ones, or easier ones, is this elbow plot, which is quite fast to compute. So let's compute this elbow plot, and as you can see, we obtain a graph showing the standard deviation for each PC. We can fix up the axis, but you can more or less see that this is PC 1, 2, 3, 4, 5, and so on. And the objective of this plot is to basically figure out where do we set the threshold or the limit. So we try and find the elbow, the point where the amount of variation explained by each PC isn't really big anymore. So again, this depends a bit on your data set and where do you want to set the threshold. But for example, I would set it around PC 15, 13, 15. As you can see, after that, the graph turns into a bit of a plateau. Great, we'll finish off with clustering. Now I won't go into very much details. I will just show you the basic functions of the Seurat object. And first of all, we need to compute neighbors. So I selected here the top 15 principal components, which is what the algorithm will use to group the cells into different clusters. And as you can see here, it's computing the nearest neighbor graph. Right, so once we do this, we can find the clusters, and you can set the resolution parameter as you want. So usually what you do is compute it using different resolution parameters, and then you check which resolution parameter makes more sense depending on your data. I will show you what the difference is in just a minute. But basically, the lower the resolution parameter is, the less clusters it will group your data in, whereas with a higher resolution parameter, it will try and find more clusters. Just so you know, right now in the metadata, we also find this resolution parameter columns. For each of the resolution parameters we asked the algorithm to compute, and the numbers here indicate to which cluster do each of the cells belong. The nicest way to visualize this is using a plot. So we will just plot our PCA. In this case, I chose resolution parameter 0.1. And let's just zoom in, and you can see there are 8 clusters, and each of the cell is colored according to the cluster it belongs to. So we have cluster 0 over here, cluster 1 over here, cluster 3 over here, and so on. Now, what kind of cells are each of these clusters? Are they CD8 cells, B cells, natural killer cells? Now, this is a different step where you look into genetic markers to try and identify each cluster. But for now, let's stick to the resolution parameter. We can check, for example, resolution parameter 1, just to get a bit of contrast. And here you can see it grouped the cells into 21 clusters. And yeah, it basically depends on your data set. You have to try and check what makes sense. Now, I wouldn't go with a resolution parameter of 1 with this data set, because as you can see, you can barely distinguish between different clusters, especially over here. So it might not really make sense to subcluster these as much. But again, it depends on what you're looking for. Perhaps you are looking to distinguish a specific subcluster from the rest, and in that case you would go with a higher resolution parameter where you can actually identify that subgroup of cells you're interested in with a higher resolution parameter. Nice. So after checking all the resolution parameters, I decided to go with a resolution parameter of 0.1. It's the one that makes more sense to me. So I will set the identities of these cells according to this resolution parameter 0.1. So now each cell will be identified by the cluster to which it belongs to. So there will be certain cells with number 0, which means they belong to cluster 0, or number 1, which means they belong to cluster number 1, and so on. Nice. So another dimensionality reduction method, a non-linear one, is UMAP. Now this is a very popular method. It also seems to give better results than TSNE, but you can compute either one. For now I will run UMAP, and again I will just use the first 15 PCs. That's where I set my threshold. Right, and we can also visualize the UMAP using dimplot. So this is our UMAP plot. As you can see, it might be even nicer than PCA in this case. We can see clear grouping of clusters. And as you can see, this is only a way to visualize our data set. So each dot is a cell, and we can actually color them by the expression of a certain gene, or in this case, by the cluster to which they belong to. So the next step would be to identify each of the clusters, so to give an identity to each of the clusters depending on gene expression data. So we would look at different genetic markers for different cell types and to identify the different cell types in our data set. But this is a story for another day. And don't forget to save your Seurat object in case you want to use it later on for further analysis. You can do this easily, saving as RDS. Squidtastic. So I hope you liked this basic RNA-seq preprocessing pipeline tutorial. I hope it was useful. If you have any more questions, please leave them in the comment section down below. I will do my best to answer them. And yeah, thanks for watching. Don't forget to subscribe and like, and see you in the next one.